Well, thank you so much, everyone, for joining us. Welcome to today's StyleDB Tech Talk. I am delighted to bring our June edition. We had a break in May, and so here we are in June. My name is Devika Garg. I lead product marketing at StyleDB. I'm joining you from Northern California. I'm joined also by my colleague, Maher Hamdi, who's ensuring smooth operations. Thanks, Maher. And today we'll be talking about fundamentals in life sciences research that can make or break AI outcomes down the road. And I'm excited to introduce Jesse Johnson, who is joining us today from the Boston area. That's right? Boston in that.
Normally this week I'm in Vermont for the
Oh,
first week of summer break, but yeah, that's right. Normally I'm in Cambridge.
nice. Okay, Vermont is beautiful this time of year. And so Jesse has decades of experience in engineering and leadership roles from companies ranging to, from Google to Verily, and then from Sanofi to biotech startups like Celarity and DuPont. You're now an independent consultant, Jesse, and you help pharma and biotech teams develop practical AI solutions. Thank you
That's
so
right.
much for being here. Uh, very excited. So
Thanks for having me.
of course. So AI is delivering significant efficiencies across the spectrum. But what we're seeing again and again is that there's still a lot of groundwork that needs to be addressed in the pursuit of really getting data and infra that is actually ready for AI. So we're going to talk about that. And I'm going to be moving our slides. That's us. That's our topic for today. And before we go in, we would like to run a short poll, a couple of questions to understand where you are in your AI journey. And if you could please take the time to answer, this will be helpful as we dive in. Jesse, in your consulting roles, like how often are you seeing Gen AI popping up for scientific use cases?
Yeah, so
But
I think it's something that everyone is talking about. Everyone's really excited to do it, and and we're seeing a lot of potential, academic papers and companies that are doing it. But one thing I hear a lot, especially from larger pharma companies, is that while it's easy to insert AI into kind of operations and commercial and manufacturing, finding scientific use cases for, so like early R&D of Gen AI and ends up being a lot harder for, for some reason that no one can quite put their finger on. But, it has to do with just the fact that it's much more complicated when we're talking about scientific use cases versus operational use cases. And, scientists tend to be a little more skeptical. And of course, like some of the things we're going to talk about later in this webinar.
great. So what are we seeing? So we're seeing about like 20% on the operational side, but like a significant number in early discovery. So yeah, I think it's moving along. We have another question. Oops, a second one. Let's see, what's that? It's more relating to, problems with AI projects in production. I don't know, Jesse, I mean, do you see themes in your work?
Um, Yeah, I mean, I think a lot of the problems that I've seen are kind of extensions of things that have, I think that were there even before Gen AI became really popular. Even when we're looking at, in data, big data in biology goes at least back to the Human Genome Project. And there have always been these points of friction, especially around kind of inserting new techniques into scientific processes that have found ways of working. Bench scientists have always found ways of analyzing their data. And so when you now give them a new technique, it has to be significantly better and it has to fit into the way they want to work. And so I think a lot of what I'm seeing today is just more of that, magnified by both the potential and the extra complexity.
Hmm, makes sense. And some of the themes here we're seeing, okay, a lot of people have actually not tried anything in production, which I think is very relevant. Like we're going to talk about more about this, and then struggles with inconsistent, incomplete, poorly annotated data sets seems to be the biggest. We will talk more about that. So I think this is good. All right, great. Thank you, everyone, for indulging us. Um, a couple of housekeeping notes, actually, before we really dive in. Please add questions in the Q&A panel anytime during our presentation, and we'll tackle all of those at the end. Also, as always, we have a raffle for a $100 Amazon gift card at the end, so stick around. Let's keep going. Right. So this is our agenda for today. We're going to talk about some of these obstacles in biotech and pharma and how, metadata is absolutely critical. And then how do you really scale? Um, so Jesse, please take it away if you'd like to control the screen, or I'm happy to do it for you.
Cool. Yeah. So Um, thanks, Devika. So first of all, I think when we think about AI/ML in biotech, it's there's a lot of big problems. The kind of that we see first that people like think about, finding the places where you can add the most value. Just the fact that the data is so expensive. But as we kind of alluded to, there are let's see, and can I? Actually, I'm not sure if I can advance it.
I'm happy to.
Okay, yeah, you can go one more. So, right, so what we're going to talk about today is accessibility of metadata, and and this is something that is kind of it's kind of boring. People don't like to talk about it, but it ends up being a big problem in some kind of unexpected, possibly surprising ways. Next slide. So the way that things would ideally work, the way that we kind of want them to work is, it's often called the virtuous cycle. And so in this case, it's that the AI team or the data team starts to introduce tools. The bench team sees the value in them, sees it improving their lives, leading to better decisions. And so they do more to support the data team because often, again, because the data team's kind of outside of the bench scientist world, and so they need some help kind of getting in, a lot, especially with collecting data in ways that the data team or the AI team can use. And so when the bench team sees the value, they will make changes that allow the data team to work more effectively. That means the data team is able to get the data they need, create more value, and then the virtuous cycle continues. But what more often, at least in my experience, we see is the vicious cycle, right? So if that first time, if you don't get to a point where the bench team sees the value, they're busy, they've have tight deadlines, they have a lot of work they need to do, and so if you ask them to do something differently to support the bench, to support the digital team, the data team, it's often very hard to get them to do it, because again they have a lot of higher priorities, they have tight deadlines, and so that makes it harder for the data team to demonstrate that value because they don't have access to the data they need. And then, of course, the bench team say, well, okay, they're not doing anything. This clearly is just a waste of time. And even if they don't say it out loud, they start thinking that, and the vicious cycle just gets worse. And so, so often, I think more often than not, I've seen this vicious cycle kind of being the norm. And so the question is, how do you get to that virtuous cycle? Yeah. So just quickly, the my background, as Devika mentioned, I've done some work in outside of biotech, and then more recently some work in biotech. So I've seen this firsthand, and what I'm doing now at Miralogic is helping particularly medium and large size pharmas that have all of this data, that have this potential to use it, especially now with AI, to really to leverage that data in ways that they couldn't before. I'm working to help them turn these kind of high level AI strategies that everyone's developing now, turn those into concrete plans and successful pilots. So if we go to the next slide, what I'm going to talk about in particular is this three-step process for turning that vicious cycle into a virtuous cycle. And so we'll start with finding a convenient problem, and I'll explain what that is when we get to it. Then what I call a cardboard and duct tape prototype. And then the third step, once you've developed that prototype, to actually get the scientists to start using it, the last step is what I call catching the bus. And this, anyway, so we'll dive into what all those mean. But first, on the next slide, oh, maybe one more slide. So I'm going to go through an example. And so this is based on a project that I've worked on a few years ago. Names have been changed to protect the innocent. But so I'm going to kind of talk about it in a very high level way. But this is based on well, it's mostly one team with some amalgamation from other projects I've worked on. So imagine a biotech startup that is doing phenotypic screening of small molecules. So you have a sample, in a plate, 386 or I always forget the 15, 30, I forget the bigger size. Anyways, you have a plate with some number of wells in it. You put a drug candidate in each well, and then you stain the cells and you take a high content image of each well in order to look for specific features. And so in this case, the biologists were doing is they were using conventional image analysis where they would use cell segmentation or image segmentation, identify specific kind of human defined features of each image, and then based on that, they would calculate metrics and pick hits based on those metrics. But they found that the positive and negative controls that they were using, often the z-scores that they were getting were very small, which means that there wasn't, in terms of the metrics that they were able to calculate using this method, there wasn't that much of a difference between positive and negative controls. And so that meant that it was very hard for them to have confidence in any hits that they found. So what they wanted to do, if you move forward one more, they wanted to introduce machine learning models, image recognition models, in order to see if they could basically find better ways of distinguishing positive and negative controls, and therefore find better, more confident hits. And it turns out from purely theoretical perspective, this actually isn't too tricky. In fact, you can use models that are good at general image recognition. They actually do pretty well with cells as well. So if it can identify the difference between cats and dogs, with a few tweaks, it can often identify these phenotypes in cellular images. Bit surprising, but it turns out it works. And I should also mention that this is for a kind of conventional machine learning model. This is before ChatGPT, and in fact, so it actually makes it a little simpler. And I'll talk at the end about how this gets more complicated when we get into kind of what we call AI today with LLMs and foundation models. So if you go to the next slide, what the biologists, the bench scientists were doing is in the lab, they were creating these plates, the physical plates. And then as they were doing that, they were keeping track in Excel of what was in each well and each plate. So, everything from incubation time and cell type, which, typically would be for the entire plate, down to individual wells, what compounds were in there and concentrations, things like that. So then they would run the plates through the digital microscope. And then on the next slide, they would then manually take the information in the Excel sheet about which compounds were in each plate, and they would merge that, join that with the features that they found in the images, and then based on that, they would figure out what were the compounds that actually gave them the hits or not. So now they wanted to introduce, thanks to these machine learning models, and so they were using, Python scripts, Jupyter notebooks, things like that. And so they needed the data to be in a fairly standard format in order for these scripts to run as is. And so that was fine for the data itself, that is the images, because the microscope output those. And of course, digital microscope didn't actually look like that, but it output the images in standard formats, in a standard file structure, file names, all of that. And it took a little bit of technical work to get that to a place where the machine learning team could use them. But in the grand scheme of things, it was not easy, but at least straightforward. The harder part was getting the metadata, so that is the information about what plate is in each well. So in order even just to know which were the positive and negative controls, so they could build their models, and then figuring out which compounds were corresponded to an image that was a hit or not. So if you go to the next slide, what they ended up having to do was this manual cleanup step, because every time a bench scientist ran an experiment, they would make the Excel file a little bit differently. They would use, different column names. Often there would be special aspects of the experiment that needed an extra column, like maybe they were doing, they were doing some kind of intermediate application so they needed to keep track of two time points instead of one, or yeah, things of that nature. So these Excel files were already always in a slightly different format, and so the data team, the machine learning team, ended up being the ones that had to understand, well, first of all, find where they were, get the bench scientists to email them, or put it in a shared folder. Then they had to figure out what was actually going on, interpret it, and then they had to actually clean it up and get it into the standard format. And so if you go forward one, um, that was taking them a lot of time. I think it's pretty standard for computational biologists or data scientists to spend half their time just cleaning the data in biotech, and that was definitely happening here. And then the uh, the next problem was that that meant that they were so by the time the bench scientist was ready to send them the data, they had already run them through the microscope, and they'd probably done the conventional analysis themselves. Then they had to tell the ML team about it, and then they had to go through this process, which could take days, and then they had to run the models, give them the results, and so it could easily take a week from the time the experiment was completed until they actually had results. And by that point, the bench scientists had typically kind of moved on to the next experiment. They didn't care about this anymore. They got good enough results. They were fine with that. And so even though the machine learning team was generating results, they were effectively not being used. And so I think maybe I am able to, yeah, I think I can advance the slides now. Um, and so the ideal solution, right, would be to give the bench scientists a place where they could enter the plate maps in some fancy UI, maybe not a phone app, but at least like a desktop app, so that it would be capturing them in a consistent way. And then as soon as the experiment was done, the ML team would have them, and they could run their models, and it would just magically work before the bench scientists even had time to do their own manual analysis. The problem, of course, is that, well, it's a big investment to build an application like that. It's really hard to build something that is easier to use than Excel. That's why bench scientists use it. And then even if you could build something that was actually, easier to use than Excel, or even comparable to it, you'd have to teach the bench scientists how to use it, convince them to take the time to learn it. That's a big investment for them. And, they're now in a situation where they've seen the machine learning team, kind of just being slow and failing to make an impact. And so it's going to be hard to convince them to do that. Right. And so this is where we need to get into this three-step process, right? We're now in the vicious cycle. We need to do something to get us into the virtuous cycle. And so the first thing is to find a convenient problem. And the reason, and so a convenient problem is something, so basically it's a problem where the solution moves you in the right direction. So helps you capture the metadata in a consistent format in this case. That it's a problem that the bench team, so the team that you need to get buy-in, they need to feel this acutely. So it needs to be a problem that they really care about. And in this case, getting better, getting a better model. That's something they cared about, but it wasn't exactly acute, right? They were doing okay with what they had. So it wasn't quite an acute problem. But most importantly, number three is that it's straightforward and quick to address. And this was clearly not quick to address, because that's why we were getting into that vicious cycle. And so what we need is a convenient problem. And so in this case, there was a convenient problem that was sitting there, but it didn't quite make it onto the diagram yet. And this was automation. So the bench scientists, of course, they weren't using pipettes to fill these plates out, at least not most of the time. Usually they were using a liquid handler. And so they needed a way to get the information from their Excel sheets about what would be in the plate to turn that into instructions for the liquid handler. And this was a big, painful process. They had Excel macros set up. It was just a mess. And this is something that they feel acutely, because I mean they have to do it. They can't get around this. And if there isn't a good process for it, then it's hours of work for them. And so this is something that we need to solve for them. This also is a good convenient problem because, as you can see on there, if we can get them to it involves metadata. If we can get them to get the metadata into the liquid handler in a standard form, we could probably extract that in a way that will be a standard form for the machine learning team. And so this is now great. If we can build this app that lets them enter their data and have that feed the data into the liquid handler, we're good. But the problem is that we need this problem to be something that we can solve easily and quickly. And as we already discussed, building that app is going to be expensive and risky, and maybe impossible if our goal is to get it to be as convenient, as easy to use as Excel. But luckily, oh, and so this is where the second step comes in. And so in order to turn this into an actually convenient prototype where it's easy to solve or quick to solve, we need to do what I call a cardboard and duct tape prototype. So obviously cardboard and duct tape is a metaphor. We're not actually going to use those, but we are going to do is use the simplest, most straightforward tools that we have. And in that case, that's Excel. So the scientists, the bench scientists are already using Excel. Um, they're just kind of jumping through hoops to get those formats into something that the liquid handler can use. And what we're going to do is now instead of building an app where there's a user interface that they have to enter all the information, we're just going to write a very simple script that takes those Excel files and just converts them into the automation files. Because that's something that can be written maybe in an afternoon or two by a good software engineer. It's not, and it doesn't need to be, you don't need to have that fancy UI. It doesn't need to be easy to use. Um, Now, there is one little kind of trick here, which is that now the scientists have to, in order to use this script, they now have to have that data, their plate maps in a standard format. But that's okay because they immediately see the value of that, right? So they're now motivated to put it into that standard format because that saves them the hours of fighting with Excel macros to get their automation scripts. And so before, of course, they had motivation to put it in a standard format, but it was this kind of more long-term, this more nebulous benefit of getting better hits, which they hadn't even seen if that was the case. Right now, they have an immediate value from that. It's kind of a small cycle. And so this is something that we can do relatively quickly and begin that virtuous cycle because once they do this, now we can have the metadata going into the models and we can actually see if the models are better than the way that the bench scientists were already doing it.
Jesse,
Um,
can I ask you a question here?
sure.
So, where do ELNs fit in this workflow,
Mm. Great
right?
question.
Yeah.
So it depends on the ELN. Most ELNs, or at least the way that most teams use ELNs, they don't have great plate map capabilities. Now some do, and some are better than others. But in my experience, a lot of teams don't end up using the ELN for plate maps, even when they do have those capabilities. Um, Partially, it's because Excel is really easy to use. It's what they know, and so it's really hard to kind of move away from that. And again, like it's very hard for an ELN to have an easier interface than Excel. And so when you get into more farther down the pipeline, you get into regulated, the kind of quality controlled environments. There tends to be more of that because you have the regulations, but especially in early discovery, where things tend to need to be a lot more flexible, and there isn't that kind of regulatory component. It tends to be Excel. But it's a great question. And and in particular, one thing that when you start doing something like this, where you're kind of encouraging the bench teams to start adopting standard formats and things like that, that also you can then use that to actually have the plate maps in the ELN in a standard format, which again, many teams aren't doing today or wouldn't do if without having that motivation. Yeah, but I mean, that's certainly when I kind of started working in this field, I just assumed that all this stuff would go in the ELN, but it tends not to. Um, yeah. So when in the situation in the team that this use example is mostly based on, when I started working with them, they actually already had this app that allowed them to you know uh, get the data from the plate map, the pure plate map, into the automation tool, but they weren't actually using it. And there were a lot of reasons, some having to do with politics. But I think the main reason, and the reason that even though we already had that tool, that it still took some time to get the bench teams to start using it is because we needed to do the third step, which is catching the bus. And what I mean by this, and again, metaphorical, if you're trying to catch a bus, a real bus, let's see, we'll go forward. Um, Actually, Davika, maybe you can. There we go. So the idea is that if you're actually trying to catch a bus and you're maybe standing a block away from the bus stop and you wait for the bus to pull up to the bus stop and then you start walking towards it, by the time you get to it, it will have, pulled away and you will have missed the bus, right? So you can't wait till the bus is at the bus stop before you try to catch it. You have to plan in advance, look at the bus schedule, see when it's leaving, and then you start walking and you give yourself enough time so that you can get there before the bus arrives. And so what we were seeing in this situation was that the bench team, so they had the idea for the experiment, they would do all the work, generate the plate maps, run them through the microscope, and then when the data came out of the microscopes, that's when they would go to the machine learning team and say, hey, can you help us, you know, analyze the images, find the hits. And of course, by then it was too late because, well, they had already created the plate maps in these non-standard formats. They may have made some other experimental decisions that would be problematic. I know, like anyone who's worked in computational biology has seen this problem. But for all these reasons, they had basically missed the bus. It was too late to get them results in a meaningful way on a meaningful timeline. And so what we had to do was kind of be a little bit more disciplined about this and say, okay, we missed that bus. We're not going to try and catch up with that. And we're going to instead put our energy into catching the next bus. So look at the schedule, which in this case means, oh, you can go back one. Um, so in this case, find out when the scientists are planning their next experiment, what's the next experiment that they're still in the planning phase, and where it would be, it has the kind of the right context to use this. Maybe it's simple enough, it's sort of experimental enough that they kind of want to try something new. Maybe it's tough enough that they don't expect the z-scores to be very good. So find that next experiment, and then instead of trying to chase after the one you already missed, we're just going to start planning and show them how to, put the Excel sheet into the right format so they can use the automation tool and so on. So in general, I think, and again, it takes some discipline to do this, but it's really important to be planning in advance and kind of start coordinating as early as possible with the bench teams that you want to support to make sure that when the right experiment comes up, you've already planned for it and you can incorporate as the bench scientists are planning for it, they can incorporate the machine learning team or the AI team or whoever it is into that planning process. Yeah, so once we did that, and we were able to actually start using these machine learning models in experiments, and sure enough, we saw that they were able to pick out much more subtle phenotypes, and we started being able to call hits, validate those hits alongside the hits that came out of conventional methods, and sort of really see the impact of these models. Yeah. Okay, so what about AI? I think that's the question, right? Because again, this is the example that I just went through is in a fairly simple context where they were just looking at one experiment at a time. If your goal is not to build, single like hit calling models like they were, but to build foundation models or any kind of machine learning model that looks across multiple experiments, it just gets more complicated because now you've got all of these, subtle differences between the experimental methods, these fundamental differences that need to get incorporated. And so it becomes all the more important to capture that metadata in a standard way for every experiment so that you can start trying to reconcile it, and then get it into a database that can handle that kind of flexibility. This is one of the reasons that I really like TileDB because it's both like it gives you that flexibility, but also gives you that scale. And start thinking about kind of what does this high dimensional data look like where you're getting different readouts from different experiments, but you want to be able to pull those all into the same model. Um, yeah, and maybe I'll just quickly like the reason that I started using TileDB for a number of projects was because it's kind of low overhead. It's very easy to get started, even with the open source version. You don't even have to sign a contract to start using it. Gives you that flexibility. It doesn't have the UI of Excel, but if you want to take Excel sheets that are, in fairly non-standard formats and push them all into TileDB, it gives you the flexibility to do that and then kind of clean it up later. Yeah,
That's great. I mean, I think, and you were at like one of our customers is Celerity, and you were at Celerity when you picked TileDB as a
right.
solution for the single cell data. Um,
Yeah, yes, exactly.
I mean, I think this is very interesting that yes, we are using still Excel sheets and we need to at some level. And but it doesn't work at scale, is what you're saying.
Mhm.
And one thing I want to understand is like the part about like what you said that need to use databases. So why aren't there databases that like biologists can just use immediately and but they haven't been? And so if you can touch a little bit on that.
Yeah. Yeah, and I think there are starting to be more like kind of biology aware tools available. I think the main obstacle is, well, besides the fact that Excel is just really easy to use and it's what all the, bench teams know, there's also the fact that every experiment is a little different. And of course, like the farther down the pipeline you get, the more standardized they get. But even when you get into like high throughput screening, where in theory you're doing the same experiment every time, there's still changes, right? Like you might start getting into drug combinations where you're looking at multiple small molecules or multiple proteins or whatever you're screening for, in the same run. And so now these kind of traditional relational schemas tend to be very brittle. So you say, okay, well, every experiment we're going to have incubation time and a compound and a concentration, and then and that's it, right? And then the bench team needs to run an experiment where they made some minor tweak. Again, now there's two incubation times because there's some step in between them, and so now we need another column for the second incubation time, right? Or and so typically as soon as you decide what your relational schema is, the next experiment is going to break that, right? And keeps breaking it. And if you sort of just keep adding columns, then you end up with hundreds or thousands of columns, which in some cases that might be the right way to model it, but relational databases just aren't built for that. And so some things, so again, like with TileDB, where you can use sparse matrices where it's very easy to add columns for some of the data, and without having to change the schema for every single row. Now, of course, there's still complexity to manage. In some ways, this is kind of pushing the complexity to another part of the system. But the fact is that if, biology requires a level of flexibility that traditional databases aren't built for.
I think that's very interesting. And I mean, this is true of not just high dimensional multi-omics data, it's true of any type of biological experiment because you're constantly adding there's more samples and there's no and it there's another term that's thrown around a lot, like data harmonization. And how does that relate to, what does it really mean? Why is it so hard? If you can touch a little bit on that.
Yeah, and so I mean, so data harmonization can mean a lot of different things, So like in the multiomics world, one of like using the same ontologies or vocabularies is really important, right? Like if you're having different ways of naming genes, for example, that can cause problems. And so like one thing is just making sure you're using like same names for things. Then there's column names, right? Like if sometimes they capitalize the column names, sometimes they're lowercase, right? Like or they use abbreviations, things like that for the same column. Or in some cases they may be kind of encoding the same information in a way where it's not exactly like a one-to-one map between columns. For example, like you might um, uh, like if you're talking about concentration, you could do it as two columns where one is a value and then the other is the unit, or you might put them both in the same, where you have the a string with the value in the unit, or maybe you just say it's always going to be micromolar, and we just use a value there. So like there are so like there's a lot of different ways of encoding the same information. And so if you don't kind of proactively try and get the bench teams to do it in a standard way, they're not going to, just because it's kind of arbitrary which standard you use, and so everyone's going to pick a different one.
Yeah. And I think like in a lot of like our conversations also, we see this tussle between bench teams and data and AI teams, where data and AI teams, they're exactly saying the same thing, that we're involved too late. And now this data, like we can't really use it in
Mhm.
any kind of model training, but just because we don't know, like so much of this stuff is missing, like either we don't have sample tracking, lineage tracking, or like some key experimental details are missing from this part where maybe they're stored somewhere else. And so the matchmaking hasn't happened enough. So any kind of normalization, standardization cannot happen for us to use this. Um, yeah. Um,
Yeah, and so I think I've got a lot of thoughts on that. I think one thing is that the mistake that a lot of technical teams make is they try to solve the problem all at once, right? And so they say, okay, we're going to go and build the system, and it's going to take us 3 months, and then everyone's going to switch to it, and it's magically going to work, right? And then so now you have 3 months more of like messy, unorganized data, and then you have the system that hopefully the bench teams will use, but often it's, there are small snags, there's reasons that it's not going to work, probably not flexible enough, or it's, harder to use, slower to use. And so that's why I think that this kind of incremental approach, starting with a cardboard and duct tape prototype, is much likely to get, you're never going to get 100% of the way there, just because especially early discovery, um, biology is messy, but you can make incremental progress, and kind of see where the snags are as you build, rather than trying to build something and, expecting it to work perfectly. So like with the Excel sheets, if you can get the bench teams to put their use standard column names and, other conventions, because they have reason to, for automation or for whatever reason. Once you can at least get them to use a standard format, then it's easier to build the UI where they can enter it directly, right? But you wouldn't know how to build that user interface until you can get them to actually use a standard format for Excel, right? Because you give them the format, and then inevitably within a few weeks, or maybe sooner, they'll come to you and say, well, I can't use the standard format because I need this other column. And great, we'll add that column to the standard format, because it's a lot easier to add it to an Excel template than to go and update your database and your user interface and all of that, right? So you can iterate a lot faster, um, get it to a place where it's as stable as it's going to be, and then evaluate whether it makes sense to build the long-term tool.
right. And I think we're getting questioned that like if you're keeping this type of data in Excel for so long, then are you worried like won't data stay messy for super long? So how do you get it out of that Excel loop essentially into
Yeah,
like
it's a
of
great
a long
question.
term, yeah, long term
Yeah,
system?
and I think, yeah, and so I think what it comes down to is, you as you go down the pipeline, right, like early discovery biology kind of is fundamentally just like it's changing a lot. It's very hard to kind of lock it down. The farther you get down the pipeline, the more stable it gets, and the easier it is to start like even trying to collect data in a consistent way. And so I think Excel should always be used as kind of a bootstrapping, like a way of prototyping things and getting them to a point where you understand how stable they are and understand what the potential is to get that into a database. And so, so I think when you're in that zone where it actually makes sense to build something a little more stable, Excel should be the prototype that tells you how to build that thing. And when you're in the zone where it just doesn't make sense, it's all going to be Excel sheets. That data probably, like, there probably isn't a solution that's going to allow you to capture that data consistently. And so using Excel as a bootstrapping kind of step at least helps you understand like where that what your limitations there are. Um, yeah, but the goal is always, in the zone where you can, to eventually get it into a database, get it into a consistent form that you can use, to train the foundation model, or train the AI model across all of your different all the experiments that are consistent enough to train on.
Absolutely. I mean, so we're really essentially talking about getting to a point of AI ready data, which is like a very key part in CIO and CDO agendas today in general.
Mhm.
Like even like heads of R&D, they're like, I think everyone is kind of aligned that, yeah, how do you get your data to be AI ready? When you consult with biotech and pharma, what does this mean beyond what we've already talked about? Probably like from a governance point of view, data quality point of view, how do you manage the like imbalance of having high quality data but then very low quantity of high quality data? How do you, and like where you like all these models, they need a lot of data, so how do you overall talk about these things to leaders in this space?
Yeah, I mean, I think it always comes down to, like, you need to have that endpoint of, like, where you're getting real value out of it, right? Like, you need to have a reason to put that investment in, to put the data into the right format. And I think one reason that I'm excited about AI, especially as we're kind of seeing more getting away from the hype, getting into the concrete use cases, is because it changes the calculus about how data can be used. Right. So like machine learning models, or like, I guess, like before LLMs became, before ChatGPT, right? Like machine learning models were still fairly restricted in the kinds of data that they could use. They had to, use something in a fairly narrow scope. It had to be fairly consistent. As we're getting into foundation models and things like that, it's much easier to train models on lots of different modalities, where you can have a little bit of data in, slightly different contexts, slightly different formats. And, LLMs are able to actually, kind of get value from the intersection and the overlap. And so now this data that, so if you screened on a target, a particular target, and then you decided to abandon that target, in the past, like, that data was basically worthless. There's nothing you could do with it. Whereas now we're starting to get into foundation models that are able to use, the three-dimensional structure of proteins in order to say, like, okay, well, knowing what I know about, a dozen targets that we've screened in the past, I can now actually predict which molecules will work on new targets based on the structural similarity between the proteins. That you know wasn't possible 5 years ago, I guess, 3 years ago, maybe even a year ago. Whereas now we're starting with the Bolt 2 model, right? Like, they claim to be able to do that, and certainly, like, that's only going to get more accurate over time. So that's one example of how these data sets that didn't have a lot of value just a few years ago are going to have much more value. And so when you see that, the potential value of having them, curated and cleaned, well, that means that it's worth going back and doing that curation and cleaning, where it wouldn't have made sense as an investment previously.
It's really interesting. And I think you talked about this a little bit, but from you've been at tech companies, just like pure tech companies, versus like you've been at life sciences companies. And what is the fundamental thing that is different when it comes to data needs? Like why is life sciences data different and it's harder to tackle? I think like you need that mile deep understanding to actually build any kind of solution for this industry. Uh, that
Sure, yeah. Yeah, and part of it is, of course, just like right, you need to have some kind of biology domain knowledge, or at least to be able to, fake it enough to, or at least you need to have enough context to be able to work with bench scientists, if you're on the digital side of things. But I think the more fundamental problem, or the more fundamental difference, is that, so in the tech context, you're collecting data in a very controlled way, and typically in a very consistent way, right? So if you have an app, and you're collecting data from users who are filling out a form, that form is going to give you the same, schema, the same structure of data every single time they fill it out. And if you want them to, if you want to add a field, make a change, the same person who's collecting the data is also, or at least the same team who's collecting the data is also making that change. And so they can make sure that they can prepare for it, make sure it happens, right? In the biotech and pharma, especially in early discovery, the bench scientists are not thinking in terms of how can I make sure that the data from this experiment is compatible with every other experiment we've ever done, right? They're thinking about what's the best experiment I can do to answer the question right in front of me, right? Which is what they should be thinking. But that means that if the question changes a little bit each time, then the output data changes a little bit each time as well, or maybe more. And so as a result, there's this kind of this fundamental complexity, this expectation that you have to have that every experiment, the schema, the output data is going to be a little bit different.
Makes sense. Yeah. Um, Jesse, I think we're almost coming to a close, but any thoughts, like what would be maybe like one piece of advice and anything else that you would want to add um, to teams who are thinking about productionizing AI models?
Yeah, I mean, I think the thing that I've learned to always, think about is what's the right level of production for the particular task? Because it's I came in sort of coming from the tech world with expectations about what good engineering is and what like production means, and very quickly, probably not as quickly as I should have, but eventually realized that like that level of production just doesn't make sense if you're working on a small team where you tend to answer questions one off, right? And of course, like there are other teams where they're doing high throughput screening, they're doing more large scale experiments where more consistency makes sense, but it's always on a spectrum from, like the kind of the ideal, which is more like what you get in tech, down to this kind of very like every experiment is a one-off. It's just about getting the answers like getting an accurate answer as quickly as possible versus building something that's going to work the next time you ask that question because you're never going to ask it again. And so I think it's important to figure out where you are on that spectrum, where your company and your team is on that spectrum, and then really try and build for that, and not like data folks like me tend to err on the side of engineering more, and that's a really important habit to kind of break, or at least like recognize and account for.
Absolutely. Well, thank you so much, Jesse. Let me see what more do we have on the docket. We've been through Q&A. And I wanna just share TileDB Carrara, which is our next landmark product release that is coming soon. You can a lot of what we talked about, the organizing of your data and making sure that it is findable, accessible, interoperable, usable, and structuring into a schema that is usable downstream. You can collaborate, share, and analyze. So like Carrara is something that brings it all together. And we're very excited for this release. If you'd like to stay tuned, you can please scan the QR code to join the waitlist. This is coming in fall. And lastly, okay, it's raffle time, the fun part. We, do we have a
Yeah.
winner?
So the winner for this raffle is Kimberly Cottenham. So we'll be sending you an email very shortly.
Congratulations, Kimberly. We'll be reaching out to you. Well, all right. This concludes, I think this was very exciting. Thank you, Jesse, for joining. We do have, if you'd like to reach out to Jesse or to me, please, these are emails. Please feel free to contact us. Thanks again for joining. And
Yeah, thanks for having me.
of course, we'll see you again next month. Thanks.
Thanks, everyone.